threat. So it was very much in our remit to assist with this, hence why she's now giving the talk. Okay, Maggie, over to you.
Thanks, Martin. Yes, so as it says, sort of Tropilaelaps research in Thailand. So I'll be presenting tonight. I'll also be joined by Victoria Tomkeys from Ferrer Science. She's going to give you a bit of an update on part of the work we did. Dan Etheridge was also meant to join us tonight. Unfortunately, he's been called into work. I think he's hoping to join, so he might be here later, but you'll have to put up with me talking for the vast sort of duration of this presentation. And the first thing I'd really like to say is just a big thank you to BDI, B Disease Insurance. Tropilaelaps is a threat. It is something that we were concerned about, but I think the thing that sort of myself and Dan and Victoria and Paul and George, who also came on the trip, the thing that we sort of took away was just how valuable it was to us, and how much we sort of learned from the trip and from the research, and really we couldn't have done it without B Disease Insurance. So a big thank you to you all for all of your support there. So to make a start, so what is Tropilaelaps? Well, it's a parasitic mite, which means that it's an arachnid, so it's in the family of Laelapidae, and its primary native host is the giant Asian honeybee, Apis dorsata. So there are 4 subspecies of Tropilaelaps, only 2 of which are harmful to Western honeybees, Apis mellifera, and really you can only tell the difference between these different subspecies through microscopic analysis. So Tropilaelaps mercedesi, this is widespread throughout Asia, so that's one of the subspecies that affects Apis mellifera, and also Tropilaelaps clari, so this is restricted to the Philippines, but both are spreading and they both sort of pose a bit of a threat worldwide. So characteristically, so it's as I said, it's an 8-legged arachnid, has an elongated body, it's about 1.5 millimeters long, it's about 0.5 millimeter wide, it's kind of light sort of reddish brown, also has 2 antennae. So I've got a little video here that we took in Thailand, and you can see some Tropilaelaps on a larvae there, and they're with some varroa. So if I can get my pointer to work, if you just bear with me for a second. So you will see here that these, if they stay still, these bits at the front, which look like antenna, so they aren't actually antenna, they are sort of their front pair of legs. So it looks like they only have 6 legs, but actually they have 8. And you can also see how fast they are. So they're a lot quicker than the varroa mites. You can see that one there sort of running around harassing the varroa mites. So this one here is a pregnant female. I'll sort of go over how you tell the difference between them in a bit, but what we noticed was that the pregnant females seem to harass the varroa mites more than the non-pregnant ones, and so the other thing that's sort of interesting about Tropilaelaps is they reproduce more quickly than varroa, so they cause similar damage to a colony. So if you were inspecting a colony and it had Tropilaelaps in it, you would see that sort of parasitic mite syndrome and dead larvae, and perhaps sort of brood uncapping, things like deformed wing virus. But it's much more severe than varroa, so they reproduce more quickly. So that there's some research to suggest that they can mate outside of the cell, so they don't need to reproduce inside the cell, which is obviously different to varroa. And when Tropilaelaps feed on larvae, they cause more damage than varroa does. So they actually make tears into the larval wall, whereas varroa have more sort of piercing mouth parts. And by doing this damage, if the larvae do manage to sort of pupate and emerge out, they're actually a weaker bee. So really sort of quite a bit more severe than varroa is. So this is a distribution map from a scientific paper that was published in 2015. It shows the distribution of all 4 species of Tropilaelaps. So Tropilaelaps mercedesi is the most widespread of the 4. So as I said, this map was out in 2015. recent research has shown, so it's now thought to have spread as far as Iran and Afghanistan. And just very recently, the COLOS website, which does honeybee research, so they published details that it might be in Russia and Uzbekistan. So it's actually spread quite a distance. So what was the impetus behind the trip? Well, Dan and I, we started in the National Bee Unit on the same day, and we've worked for the NBU for 10 years now. And Dan and I, so we've both worked in the United States with honeybees, and we've seen small hive beetles in colonies there. And this sort of seeing small hive beetle made us realise that seeing something in the field and how it interacts with honeybees, it's really sort of very different to reading about a pest or watching videos on it. So that first-hand experience, we both found it really valuable. And we also had concerns about how effective our detection techniques were for Tropilaelaps when we went out inspecting in the field. Beekeepers had concerns that some of the methods we were using to find Tropilaelaps or to look for Tropilaelaps might damage the colonies. And when we did these techniques, a lot of the time we didn't see Varroa, and we knew that there was Varroa in the colonies, and that really made us wonder, well, if the technique doesn't detect Varroa, then how likely would it be to detect Tropilaelaps? Dan had also done a bit of a sort of unscientific experiment at home. He'd collected lots of Varroa mites from his colonies, and he kept them in a jam jar, so without any form of food. And they survived for about 2 weeks. And this is kind of different to the accepted knowledge of how long Varroa can survive. And again, this really just made us question, well, if Varroa can survive for this long and no one really expects them to be able to do this, what about Tropilaelaps? Does that same sort of thing apply to Tropilaelaps? So really, we just had a lot of unanswered questions. And also sort of things like, well, you know, would the laboratory sampling pick up Tropilaelaps if we sent samples in? So here we have a photo. So this is a photo of some Apis florea honey. It's in a box, and the honeycombs, so this box of honeycombs were intercepted in the UK by, I think it was customs that intercepted them. So this photo here is an Apis florea comb. So that's the red dwarf honeybee. It's native to Thailand. And you can see at the top here, you have where these bees store their honey. And then down below, you have the brood section. So you can see the worker brood there. And then down here, we have the drone brood. And when sort of people go out and collect this honey, so it's very valuable. It's believed to have a lot of medicinal properties. It sells for a lot of money. So that they'll go out and they'll collect these colonies, and they'll cut across the bit here to remove the brood. Very often they'll eat the brood, so they'll eat that as a protein source. But this honey here on the stick, so this is what you can see in this box that have been sort of sent over to the UK. And the thing that really alarmed myself and Dan about this was you can see up here that the brood is incredibly close to the honey and where they actually sort of cut across it. And potentially there could be Tropilaelaps in this brood. So potentially this that was posted over could have had Tropilaelaps in it. And one of the questions was, well, would this survive? Obviously there weren't any bees looking after that brood, but could Tropilaelaps survive for that long to come over to the UK? Yeah. So lots of shipping containers enter the UK each year. Last year we were called out to an Apis florea colony, so that was on the side of a shipping container. It had come from India. Now the colony was destroyed, but there was comb and there was live bees, and again that there could have been Tropilaelaps there. Vans and cars, they're regularly stopped at ports with all sorts of things in them, from queens and attendants that are being illegally imported, to equipment, drawn comb, full-size colonies with bees actually flying around like in the car. We've had that before. And we don't always know where they come from. So generally they come from Europe, they sort of turn up on a ferry, but we don't know the point of origin of all of these things. And the other thing is we don't really know how far Tropilaelaps has spread. So as I said at the start, we know it's made it as far as Iran, but could it have made it into Turkey? Nobody's sort of really sure. We don't think it has, but nobody's kind of 100% certain on that. And everything I've spoken to you about just now, they're the interceptions that we know of. These are the things that we get told about. But there's probably quite a good chance that there are other things that are reaching the UK that without our knowledge that we just simply don't know about. So we had all of these concerns, and Dan and I, we both felt that it was important to try to address them. And the only thing we could think of doing was to visit somewhere that actually had Tropilaelaps in colonies, which would mean going to Asia. So I knew that the NBU wouldn't have funding for something like this, but it was around the time, it was the BBKA news, and I saw an advert that BDI had placed, and it was asking for applications for research funding. And I thought, you know what, it's worth a shot. So this is a bit of a sort of bullet point of the objectives that we put in our application. So this was everything that we wanted to achieve. But the main sort of headline objectives were that we wanted to see Tropilaelaps and gain experience of what it looks like, how it behaves, and to see if our detection methods would work. So we thought Thailand would be a good place to do the research. So Panawan Chanwanakul, so she was based at Chiang Mai University. Unfortunately, she passed away early last year. But prior to her death, she had done a huge amount of honeybee and Tropilaelaps research. And I knew that Chiang Mai and other universities in Thailand, so they were producing lots of good Tropilaelaps research. We costed everything out. We had a budget of about £7,000. We thought we could do it for that. We had a bit of a contingency fund, and we thought if we could send 4 inspectors to Thailand for 2 weeks, we could achieve all of these objectives. So I submitted my application and was really thrilled when BDI came back and approved it. But then it was also slightly scary because we actually had to go and do all of these things and sort of put our money where our mouth was. So first thing Dan and I did was put together a team to go to Thailand. So we have Paul Davies here and George Tong. So they're 2 seasonal bee inspectors from Wales, and they were chosen. And Dan and I also redid the budget at this point, and we realised that with the contingency fund, we didn't have enough money to bring a 5th inspector with us, but there was going to be a reasonable amount of money left over. And we thought that it would be beneficial if someone from Ferrous Science Limited, so Ferrous Science do all of the diagnostic work for the National Bee Unit. So if you have foul brood or something, the samples are sent into the lab, and it's Ferrous that does that work. And we thought it would be really good if they could send somebody and we could trial the laboratory detection methods as well. So I spoke to Ferrous on the off chance. I thought, you know, maybe they might have a bit of spare cash. Fortunately they did. And so Victoria was able to come with us. So we had our team, and then the planning began. So this was one of the planning meetings that we had in North Wales. So Victoria lives in York, Dan's down in Hampshire, and myself, Paul and George are sort of spread around Wales. So we all met up in various places to plan and go over things. So the first question was, where will we go and whose bees will we see? So I started putting out feelers. I spoke to some of the honeybee researchers I'd worked with in the past, and they put me in touch with Bajri Chushul of Chiang Mai University. She had taken over from Panawan when she passed away. Kittifong Konfinit Bunjong, so he's from Mae Phau Lang University. He had been Panawan's PhD student and postdoctorate assistant, and Guntama Swanapong from Burrup University. So she works with Samuel Ramsey. She also does a lot of Tropilaelaps research. So from left to right, we have Bajri, so also known as Bomb, Kittifong, who's known as Golf, and Guntama, who's known as Gun. So Gun is probably the only beekeeper that I've met. So she went out inspecting in these 4-inch heels and the dress, and it was about 30 degrees heat, and we really did go out and we looked at a lot of colonies, so that was quite impressive. So the next issue and the biggest part of the planning were, what were we going to do in Thailand? We had these objectives, but we only had 2 weeks to meet them. So how do we efficiently do what we sort of set out in the proposal? Well, number one was we added an extra week to the schedule. So we had enough money in the budget to do this. And this gave us 2 weeks of field trials in Chiang Mai, and then 3 days at Burrup University, plus some traveling time to get there. So the next thing we did was we dissected our standard operating procedure. So everything we do in the National Bee Unit everything that Victoria does has a standard operating procedure, or SOP, and we'll talk about that quite a lot. So these set out how we do things. So this SOP has been updated now, but before Thailand, these were the 4 detection methods that bee inspectors used in the field to detect Tropilaelaps. So you're not bee inspectors, so I'm going to briefly go over what we originally did before we went to Thailand to look for Tropilaelaps. So the first method, examining brood cells and comb, and for this we would take an uncapping fork, and ideally we'd fork out some drone brood that was at the pink-eyed pupal stage, and we'd look for mites on what we pulled out, and the SOP specified that we should do this to 200 cells. So you can see there's some varroa here on the pupae that have been pulled out. And the theory was that, you know, we would see Tropilaelaps in the same way that you can see varroa there. So that was the first detection method. So the second detection method was floor scraping. So this is a varroa floor insert with some hive debris. And for the second detection method, we would visually examine this board and we'd look for Tropilaelaps out in the field. And then we would scrape all of this debris into a bag and we'd post it to Ferroscience, and Victoria and her colleagues would then screen this debris for small hive beetle and Tropilaelaps. We also have voluntary central apiaries where beekeepers collect and submit floor debris samples 3 times a year to be monitored. So I will show you what Victoria did to those samples in a second. So the third detection method is the comb bump method. So this is where you take every frame of brood from a colony and you bump it on a white tray to dislodge phoretic mites. And we would do 2 bumps per side of the frame. So this is a video that we took in Thailand. It shows the bump methods. And here we're doing 4 bumps per side. So I'll come back to that later. But one issue that we had identified before we went to Thailand was that beekeepers really didn't like us doing this method in the field, because as you can see, it's quite damaging. You know, we're banging the frames quite hard, and you know, people weren't sort of terribly happy when we did that. And then the fourth method, so this is using inserts. And so we've never actually used this method. So this method is only supposed to be used when and if Tropilaelaps is detected. So it's an advanced surveillance method. But if we ever did detect Tropilaelaps in the UK, seasonal bee inspectors would be deployed to the area surrounding where the Tropilaelaps was detected, and they'd be sent out with miticides such as Apivar and sticky floor inserts, and they would use the varroa treatment on the colony, and they'd insert the sticky floor, and then they'd return 48 hours later and inspect the sticky floor for Tropilaelaps. And the theory behind this method is that the miticide would kill the Tropilaelaps. They would fall onto the sticky floor, get stuck, and then the bee inspector would be able to see them. And by doing that, we would know how far Tropilaelaps had spread. So this all sort of seems quite sort of logical and sensible. So that was the field work side of it, and how we inspected for Tropilaelaps before we went out to Thailand. We wanted to test the efficacy of those methods, but we also wanted to test if Ferra could detect Tropilaelaps in hive debris. So this is a video of that process with Victoria.
This is a sample that we collected from the floor of one of the Thai hives, and we brought it home. So what we're going to do is tip the entire sample into the top sieve. I'll make sure that we get every single bit of debris out of there. But to demonstrate what we do, we then pop the stack of sieves into the sink and then wash the sample through with water. So we have an adapted shower head that gives high pressure water. We just wash it through. See here, we've got grass, bits of old wax. So what I'm going to do now is remove the top sieve pop it into the tray so we don't lose anything. And I'm just going to now do this sieve just to make sure that everything we want to wash through has washed through. So only things at 1 millimetre size or greater will be trapped in this sieve. And this is the 0.7 millimetre size sieve. And the 0.355 size. So if there were any trop, this is where we would expect to see them. Obviously have to be very careful that we don't allow the sample to go down the sink. So making sure that the sieve doesn't get full with water is important. And allow it to drain. Now, ordinarily, what we now would have done is floated the.
So I've stopped the video there because there's a bit of a spoiler at the end, which we'll come back to later as well. But that's the process of when the floor debris samples were sent in. That's what would happen to them in the lab. So we had our SOP. We knew the methods we wanted to trial. But we felt it would be also good to trial some other Tropilaelaps detection methods. We wanted to see if methods that are used in the UK for Varroa detection could detect Tropilaelaps. So we decided to try an alcohol wash, CO2 monitoring and an icing sugar roll on colonies while we were in Thailand. So Vita Pharma kindly supplied us with some Varroa easy checks. So they're there on the left, sort of little shaker pots there. And we adapted one of those to be able to accommodate the CO2 valve. While we were kind of preparing and that, we also found out that Thai beekeepers use Langstroth hives, but they attach the floors to the brood bodies. So you can see here that it's just sort of attached onto the colony. So this was going to make collecting floor debris and using the sticky floor inserts potentially a bit of a nightmare. And you'll see at the top here, we made a very shallow wooden frame that was covered with a thick plastic mesh. And these were to stop the bees from accessing the sticky floors that we planned to put in the colonies. So we'd done all of our planning. February came around and we were off to Thailand. We were very excited. So we travelled for about 38 hours to get to Chiang Mai. It's in the north of the country. It's an amazing city. It's really beautiful. We had Saturday and Sunday to get over jet lag, but come Monday morning, we were there to work. So off we went to the university. And Bom and everybody at Chiang Mai University were really helpful. There'd been lots of emailing back and forth beforehand, but really they didn't know us at all before we went over, and we couldn't have asked for better hosts. They were so helpful. So this is the team. So we have Yam here, who was a master's student, and Ying, who was a PhD student, and Ying turned into a bit of a fixer for us. So basically anything we wanted, Ying was able to sort out for us, and she was fantastic. And here you can see Victoria and I hugging some boxes. So we thought that we would be really organised and post all of our equipment before we left, so that we didn't have to take it on the plane with us. Turns out that Thai customs really don't like it if you post Heston Blumenthal kitchen scales to Thailand. So Thai customs seized our boxes. We had to pay about £300 to get the boxes released from customs. So these scales are now officially the most expensive set of kitchen scales in the world. But we needed the kit. We posted it all over, and everything kind of relied on the fact that we had this kit. So and it was literally released on the day that we arrived. So there was a big panic beforehand of checking the DHL website to see if our boxes had been released or not. So this is a photo of Don. Oh, if I get my mouse, so that's Don there. And he's a beekeeper that we met at one of the street markets over the weekend, and we got chatting to him. We asked him about his honey and his other products. And it turns out that Don was the beekeeper that BOM had contacted, and he'd made all of the arrangements with the other Thai beekeepers to source colonies for us. So it was one of those bizarre chance meetings where he said, "Oh yeah, I'm working with some researchers from the UK," and we said, "Oh yeah, that's us." So BOM had given us an office and a laboratory to use while we were in Chiang Mai. Don had organised bees and apiary sites. Here's a map of those sites. You can see we did quite a bit of travelling. So the furthest apiary up here in the north, that's about a 90-minute drive from the university. So we were travelling around Thailand quite a bit. And we spent the first morning setting up the lab, unpacking the boxes, just really sort of finding our feet. And then after lunch, we headed out into the university apiary. Initially, we just wanted to see some Tropilaelaps. None of us had ever seen one before. And we were in the apiary for probably about 4 hours. And in that time, with the 5 of us there, we only found 2 Tropilaelaps. And this is the first trop we found, which we named Terry and stuck it in a pot because it was so difficult to find him or her. It was such an event. So the thing that really frightened us, though, was how impossible it was to find the Tropilaelaps. We knew that they were there in the colonies. We'd started uncapping brood, we were doing the bump method, and we just weren't seeing any. So we also realised really quickly that when you look in a colony and you start doing this sort of work, there's lots of really tiny brown dots in a beehive, and it's incredibly difficult to tell if that dot is a Tropilaelaps or just a brown dot. So at the end of the day, we were all sat around the table in the office, and I was really concerned that this whole project would be a waste of time and that we wouldn't actually be able to achieve anything because we just couldn't find Tropilaelaps. And I was really sort of quite depressed and down at this point. But we had beekeepers to see the next day. We weren't going to give up. We needed to trial the salt, but we'd gone there for a reason. But we just sort of realised that really it was going to be a bit harder than we initially thought. And this is what we'd gone to Thailand to find out. So this was the whole point of it. So we sort of rallied and off we went to our first beekeeper. So this is one of the apiaries we went to. It's a fairly typical apiary. It's amongst the sort of generally the hives are kept amongst trees and they'll be in circles. And this is a time lapse of us doing those SOP methods sort of out in one of the apiaries. And you can see we're sort of doing it to all of the colonies there. So detection method 1, examination of brood cells and comb. So basically this was all I did while I was in Thailand. I was uncapped brood and looked for Tropilaelaps. I probably uncapped about 6,500 cells. Quite quickly identified that there were a few issues with the original SOP. So as you saw in that video, Tropilaelaps are really fast. If you pull out multiple pupae like this with an uncapping fork, the Tropilaelaps immediately move away from the light and they go into all the little crevices. So you just can't see them amongst the pupae. Also, it's really difficult to look at a frame of sealed brood and judge the developmental stage, of the brood there. There's no way of guaranteeing that you're going to select that pink-eyed pupal stage. And you really want that because it pulls out and kind of stays together better than larvae. But really, it's just a bit unrealistic to be able to see that and gauge what you're pulling out. So the uncapping fork, it also damaged the larvae, and you can see that there's sort of quite a bit of damage done here. And we found that any sort of juice or fluids that came out of the larvae, they would really camouflage any Tropilaelaps. And it became apparent really quickly that uncapping forks just weren't fit for purpose. So the solution that we came up with was to individually uncap. So this is a rhombus that we made. So to individually uncap 100 cells using tweezers, and I'd pull out each pupae or larvae. I'd visually examine it for mites. I'd also have to keep an eye on the cells that I just uncapped, because very often Tropilaelaps would come out of the cells and onto the comb. So I didn't do 200 cells as the SOP sort of originally specified. Like I said, we made this rhombus that shows 100 cells. But this technique is quite time consuming. It's a lot slower than just using an uncapping fork. In the end, I probably got down to about 10 minutes to do these 100 cells. But we did find out that by being more precise with it, and by just looking at the 100 cells, we did reliably detect Tropilaelaps in a colony. So We have no proof, but our feeling was that Tropilaelaps doesn't preferentially infest drone brood. And really logically, there's no reason for it to prefer drone brood because it reproduces more quickly than Varroa. So the stage of larval or pupae that you pulled out of the cell seemed to affect whether the mites remained in the cell or whether they were visible on what you pulled out. So if you uncap a larvae and you pull it out, the mites tended to remain in the cell. And this was also the case for as soon as the pupae had started to go brown and started to discolor, the mites would tend to stay in the cell. So that's like the brown one there in the photo. And I only really saw Tropilaelaps on that white-eyed or pink-eyed pupal stage, and I assume that they were sort of feeding then, but I didn't see it when the bees were any other developmental sort of age. So floor scraping. So here you can see Dan and George collecting floor debris. So this was the second method in our SOP. So we had to remove all of the frames from the hive to access the floor because of the way that the floors are attached to the hive body. So in a second, they'll put the debris in a bag, and this is what would be sent to Vera. But you can see that by scraping it up like this, it all sort of gets compacted together. And we had concerns that Victoria's sieving method might not wash the mites out of these clumps of wax and other hive debris. We're also concerned that the mites would get damaged by this scraping, so they're not as robust as Varroa. And if you remember a few slides ago, I showed you Victoria washing the debris through the sieve. So after that, Victoria would usually take those sieves and she would float the contents in alcohol. So insects and mites, their exoskeletons are made up of chitin, and when the hive debris is floated in alcohol, any Varroa or insects parts, such as small hive beetle, if they're in that sample, they'll float on the top of the alcohol, and Victoria is able to see them, but all of the rest of the hive debris would sink in the alcohol. So something that came up during the planning part of the trip when we described this technique to Bom and Kitty Fong was that they both said that the sieving method wouldn't work because Tropilaelaps doesn't float in alcohol. And initially we thought, well, they must be wrong because it's not just the UK that was using this technique to monitor for Tropilaelaps. There's quite a few countries all over the world that use the same method. And one of the first things that Victoria did was to test this and to see if Tropilaelaps would float in the alcohol. Well, Victoria did this numerous times and she couldn't get Tropilaelaps to float in the alcohol. So Bom and Kitty Fong were right. And really the laboratory monitoring that we've been doing prior to Thailand couldn't really be relied upon to detect Tropilaelaps. So I've already shown you the video of the bump method. And what we found out was that it was really ineffective at detecting trop. So we would bump the frames on the tray. It would dislodge mites some of the time, but more often than not, we would look at the frame and we'd look at the comb and we'd see Tropilaelaps running around on it, and they hadn't been dislodged from the frame or from the comb. So we were also going back into the hives 24 hours later to remove the sticky floors, and we noticed that any of the frames that we'd bumped, so it had universally killed the brood on those frames, and the bees were pulling out the brood. And what you can see here, so we bumped 2 frames in this colony, and you can see that the bees have pulled out all of the brood in that 24 hours. So in the old SOP, we had been bumping every brood frame. And when we finished an inspection in the UK, we didn't really know what happened to that colony and what happened to the brood. And as I said before, beekeepers in the UK, they're always a bit reluctant to let us do this method. So it was really good going to Thailand because it allowed us to see this damage and we also saw that bumping the frames didn't really dislodge any Tropilaelaps. So that was quite alarming that it was just so ineffective. So sticky floor inserts, these were the final methods. So here's a quick video of one so you can see what it looks like there. So, as I said at the start, sorry, excuse me, the SOP specified that we should use the sticky floor inserts with a miticide to knock down any mites that might be in the colony. So we took a range of UK Varroa treatments with us. And because we wanted to see, which one of those would be the most effective for Tropilaelaps detection. So one issue was temperatures in Thailand. So while we were there, it was between sort of 28, 33 degrees. And this temperature range, it's really outside what the manufacturers recommend that you use. So we had concerns that it might kill the colonies, or that they might abscond. So another issue was that tired beekeepers really didn't want us putting anything in their hives. But it's kind of ironic, because they were happy to use all sorts of chemicals and mite treatments. So in the center here, we have a dog flumethrin based flea treatment, and this is sort of a mister thing, and it got a bit lost in translation, but they were either spraying this on the bees, or they were spraying this on the frames to treat for Tropilaelaps. And on the right here, so this photo here is a Chinese fluvalinate treatment, which is like a cardboard strip, and this this was left on the floor of the hives for the bees to walk over, and these seem to be permanently in the colonies. And these treatments really made us concerned about sort of chemical resistance, because there were still healthy Tropilaelaps in these colonies, and that had been treated with these chemicals. We also realised that it's virtually impossible to see Tropilaelaps amongst hive debris. So even with only 24 hours of debris, so as you can see, it's not a terrible amount of debris there, but just out in the field with a hand lens, it was really hard to see them. So really you needed to be in the lab with the microscopes to be able to detect them. So one of the regrets of the trip was that we didn't get to use the existing UK Varroa treatments, because I do think that would have been really interesting to see, you know, how effective they were for Tropilaelaps detection. So that was all of the SOP detection methods. But like I said, we also wanted to trial some other detection methods. And here you'll see Dan and George using some Varroa easy checks and the 3 different techniques we did. So Dan's checked the frame for the queen. He's shaken the bees into a bowl there. George has used a half cup, just a baking measure, so it's a half cup measure, and you saw that he sort of ran his finger across the top there, and that's a sample of 300 bees, and we did that in 3 different Varroa easy checks. So that's an icing sugar one, and George is just rolling the bees in the icing sugar there to dislodge the mites. And in a second, he will show you the alcohol and the CO2 that we used.
Victoria, you might want to comment at this point about the temperament of the bees, the fact that everybody's wearing shorts and short sleeve t-shirts.
Yeah, I mentioned that, didn't I, when you were up in York? Yeah, I mean, I would never, because I work in the lab, so I'm not an experienced beekeeper such as the bee inspectors. I wouldn't go anywhere near bees without a full bee suit normally, but even I was wearing shorts. You know, they were so placid, and it's a good job because it was so warm. I think we'd have melted, wouldn't we, in bee suits, Maggie?
Yeah, it's a bit of a change going back to so even, I don't know, what is it, sort of 14, 15 degrees in a full bee suit is hotter than 30 degrees in just a hat like that. So it really was a joy to work with the bees. So yeah, George's, so this is the adapted CO2 valve that we put on the easy check. So George is just knocking out the bees there, and you'll see them sort of quieten down in a second.
They were bicycle tire inflating devices, weren't they, Maggie, that we bought in. We sort of plotted to do it here and then had to go and source them once we got to Thailand. So we bought some weird and wonderful things when we got to Chiang Mai. Yeah,
I mean, it's not really surprising that they seized our box, because there was all sorts of not questionable but unusual things in there. So you can see the bees are just starting to be knocked out now with the CO2. So like I said, you saw Victoria, you saw George knocking out the bees there. So when the thing that we do next, so we'd have the Varroa easy check, and you can see that it's got sort of a white kind of sieve bit inside it. So what we would do is we would then shake the icing sugar bees onto a tray like this. And there's Victoria with a very fine tipped paintbrush sort of sifting through that and pulling out the Tropilaelaps mites. So that was one method that we tried. We also did that with the CO2. So when the bees were knocked out and they were comatose, it doesn't kill the bees. It just sort of makes them go to sleep. And we'd shake that over the tray, and Victoria would do the same and look for Tropilaelaps mites. And then in the back here, you can see George looking at the alcohol. So the alcohol wash effectively just washed the mites off the bees. So in the UK, you know, these are used quite regularly for Varroa monitoring, and we really were keen to see how reliable they were for Tropilaelaps. So alcohol wash, it was really good for Varroa, as it is in the UK. Like I said, it's used quite widely. Not very good for Tropilaelaps. So it did detect some. We did detect some Tropilaelaps, but we think, and we're not 100% sure, but we think that the Tropilaelaps mites were just too light and too small, and that they sort of weren't really washed off the bees, or that they got tangled up in amongst the bees, and they weren't being washed through this sieve section, because you can see there's not sort of very many holes there. But for Varroa, it was fantastic. So we did find a lot of Varroa using the alcohol wash. CO2, so that's gaining popularity in the UK, and we were quite excited by this method. We thought, you know, it's popular in the UK. It doesn't kill the bees. Unfortunately, the alcohol wash does kill the bees. But what we found was that actually it wasn't very good for Varroa detection. And similarly, it wasn't very good for Tropilaelaps detection. So that was a bit of a letdown. But we were really quite pleasantly surprised that the icing sugar shake, so that was really quite effective. It picked up some Varroa, but it was better for Tropilaelaps. And what we think was happening was that the icing sugar would sort of clog up the mites. And when you had a close look at them, they'd have all sort of icing sugar under their legs, and they just seemed clogged up so that they couldn't hold on. And as you sort of saw in that video of George with the bees, all of the bees would start grooming themselves. And I think that they were really just grooming themselves and dislodging the mites, and they would then fall through this sieve and out onto the tray. And that turned out to be a really effective way of detecting the Tropilaelaps. So this is a video of Trop running around on a frame. Really, it's just to show you how small they are, how fast they are, and how difficult to see. But they do have quite a distinctive way of moving. And one thing, so as Victoria said, we weren't wearing veils a lot of the time. We were just there in shorts and a t-shirt. And obviously it was very sunny. The weather was very nice. One thing about inspecting in the UK is we really can't get away with not wearing a veil. So to see Tropilaelaps through a veil, and potentially when the light's not very good, you know, it does tend to rain quite a lot in the UK, and we'll be out inspecting and it will be cloudy, or it will be a sort of bit overcast. So to be able to see the mites in the field, ideally, you shouldn't really be wearing a veil, which isn't sort of terribly practical, but also you need that really good weather. So we were concerned about sort of coming back to the UK and how likely it would be for us to see them on the frames like this, and also how likely it would be for beekeepers, because you've got to remember that there's no bees here, and it's not terribly often that beekeepers will shake off all of the bees from their frames. So this just gives you an idea of what they're like moving around. So that was the fieldwork side of the project. We looked at 65 colonies. We did about 6 different apiaries, and we did all of the detection methods on all of the colonies. So another quick time lapse here. You can see us working, and here you can see that the beekeepers are using plastic recycling boxes. So this is quite commonly used as a hive in Thailand. So beekeeping in Thailand is it's not really a hobby. People tend to be sort of commercial beekeepers. They have from between 50 to several hundred colonies, and beekeeping is migratory. So they move the colonies around a lot to follow the nectar sources. So the hives need to be transportable, and also so they don't use supers. So that's part of what makes the hives so transportable, but instead they'll harvest honey out of the brood boxes. They don't wait for the honey to be capped before extracting it. So a lot of the honey we saw was very liquid, and so they will place buckets of honey in greenhouses to evaporate some of the moisture off. And in the middle here, you can see a Longdon tree. So that was the main forage crop while we were in Thailand in February, but they have lots of different crops that they migrate to. So things like coffee, sunflower, lychee, palm oil, rubber, all of these crops are pollinated by bees, and the beekeepers will sort of migrate and move their colonies, you know, for those crops. So the results, so to recap, using the uncapping fork didn't work, unfortunately, but using tweezers and the rhombus, so that was very effective. The screening of the floor debris samples didn't work because the Tropilaelaps didn't float in alcohol, and we think that even if they did, they sort of got clumped up in the wax there. The bump method, so that wasn't very effective at detecting Tropilaelaps either, and it was also very damaging to the colony. We didn't get to use our Varroa treatments to see if that worked. And the sticky floor, so they did work, but you had to sort of look at them in a laboratory. So as a field technique, it wasn't sort of terribly reliable. So out of those methods, we only really came away with 2 methods that we could use. And for the Varroa easy checks, we found that so alcohol was good for detecting Varroa, but not really for detecting Tropilaelaps. CO2 wasn't very good at detecting either Tropilaelaps or Varroa. And icing sugar roll was a good method for detecting Tropilaelaps. So this is a draft paper from the results from this research that we did on the field work. We're hoping to get this published shortly, and that's a bit more of an in-depth look at what we did there with more of the data. But now Victoria is going to talk to you about the other bit of the research that we did. So I'll hand over.
Hi, can everyone hear me? Yeah, it's going to be a bit like the government COVID presentations we used to have to watch, where I'm going to be going, next slide, please, to Maggie, because she's got control over it all. So while we were in Chiang Mai, one thing we wanted to look at was how long can trop survive on different matrices. So if there was ever an illegal importation and something stopped at the borders, how likely is it that trop could have survived long enough to reach the UK? So right at the beginning, Maggie showed the graph, the map of spread from 2015, and you know it was already there in India. You know, we've now been reported in Russia and various countries near Russia. So obviously it's a long way from the UK by natural migration, but if humans speed it up, could it survive long enough on an airplane journey, say from India, or somebody bringing some comb back from Turkey, that kind of thing, if it had spread over from Iran. So we just wanted to look at this in the lab. So the official title was the in vitro study. We referred to it as the pot study because we were putting bees and larvae and trop in plastic pots. But can you I've just pressed my button for next slide down there, just out of habit. So could you have the next slide, please, Maggie?
There you go.
Thank you. So the general opinion is that trop cannot survive without brood. You know, this is quite a well-reported statement that, you see that people have said that. However, a paper came out in 2021, came out of South Korea, where they sampled trop from colonies. So they sampled colonies from all over South Korea. Whilst the anecdotal evidence has been that it can't survive on the broodless colonies, the researchers in Thailand kind of, it's like, oh, well, you do get broodless periods in South Korea. So there's I think the jury's still out a little bit. They do have very cold winters in South Korea. I looked up the temperature in Seoul and it's regularly -8 in January. Kitty Fong agreed that the gentleman researcher that we met over there, he agreed that they have these broodless periods. So when we can I have the next slide, Maggie, please? If we just look at so they collected the Tropilaelaps from all regions of South Korea, the researchers in this paper. They collected from Apis cerani, which is usually in the mountainous areas, and Apis mellifera, which is the dominant type of honeybee kept in South Korea, usually in the agricultural areas. As I've mentioned, they're cold winters. And what they found was 96% of colonies contained mites. So 85% had Varroa, 76% had trop, and 68% had both. So there's a wide distribution geographically of trop in South Korea. Could I have the next slide. If anyone's interested in reading that paper, and it is a good read, I've just put the reference there. There's some quite good graphs in there as well about different viruses that are spread by trop compared to varroa. So I haven't got that, I haven't included that, but it is a good paper, and it really just makes us think, you know, all these facts that we thought we knew all this time about, you know, oh, it's really unlikely to ever reach the UK because, you know, it can't survive without the brood. Is that actually the case? Could I have the next slide, Maggie, please? Yeah, that's the one. So Maggie's already explained previously, they were so accommodating, the Thai beekeepers out there, and we were part of a good chunk of the time that we were there, we were collecting the trop because we wanted enough to set up these pot experiments. It was quite a dynamic process. We had to think on our feet. They weren't as easy to harvest as we thought. So we ended up buying frames from the gentleman in blue there in the middle. He was called Mr. Bum. So whilst Maggie and George were out doing the 24-hour follow-up inspections, and George, Dan and I were in the lab frustrated we couldn't find enough trop, they were haggling prices and bringing back frames with lots of trop for us. So certainly a good team effort. Can I have the next one? This is just a video. This is one of the first ones that we harvested for the pot, and it kind of shows how they were able to sort of move so very quickly. They were very, very difficult to handle. Go have the next slide, Maggie. So until we got our eye in to exactly what we were looking at, we were checking them all by a microscope. So Maggie sort of showed a picture of the hive debris and said, these are tiny little brown dots in lots of other tiny little brown dots. And it really was that difficult. I mean, we got better at it. We did get better, and sort of, you know, by the third day we could spot them very easily. But at first, every trop that we thought we harvested, one of us was checking them under the microscope just in case. So, you know, we just wanted to be very thorough and make sure exactly what we got that we were looking at. Have the next slide. And this is a time-lapse video of us harvesting trop and trying to get trop from the frames. The one thing Maggie mentioned about the perfect light that she had out in the field, and it was still quite difficult to see them within the lab. Head torches were absolutely essential. I mean, it was a well-lit room, and there was good daylight in there, and electric lights in the ceiling, but they were just so difficult to see that we had to have the head torches. It would have been absolutely impossible without. Yeah, thank you. So the collecting the mites, I think I would say is a lot more difficult than we thought. Before we went to Thailand, we had a Zoom meeting with Jeff Pettis in the US sort of over the Christmas, so perhaps sort of 6 weeks before we went. And he'd said it was relatively easy to collect the mites. So I think what we took from that is he perhaps had people helping him. He must have had a team of assistants helping him, or the mite level in the colonies that he was working with were a lot heavier, because it wasn't easy to collect them, and they did move very fast. The way that one of the first ways we started collecting them was with a fine artist brush, so dipped in water or honey. But if you get them too wet or sticky, they almost got clumped together. They do have a limited grooming ability, so if you get them too wet, it was quite difficult. So this video now that Maggie's got up, this just shows, so like I said, there was an adapted way that we were doing it, and we found that the best way to collect trop for an in vitro study was gently uncap the cells, and if you blow gently across them, we think it's increasing the CO2 level. They then sort of came to the surface. Mine's frozen, my video there.
Yeah, I think it might have ended.
Oh, has it?
There's another one.
Yeah, and then we just found that it was pootering them up. So we'd taken pooters with us. They were in the boxes, but they were smaller than we're used to pootering things up. So there was drinking straws and gaffer tape involved. And you can just sort of see there that I was uncapping at this time and pointing to them, and then George was pootering them all up. And they actually were quite robust to that method. There was some damage to some of them, but really it worked quite well, and we were able to get us enough that we needed to set up an experiment. And then I think on this one here, you can see we're following them round on the frame with you can just see them there following them round. So I think the cow at this point, this is with Dan's camera, and I think the lighting perhaps gives them a lighter coloration than they do actually have. Although the young ones are white, a paler in color, I think it's I think the camera possibly has bleached them a little bit. Yeah, and the next one. And here we've got, I think it was just quite nice because it's a good close-up of the trop next to the Varroa. And you can see how much more mobile the trop are, and they're certainly not shy when it comes to kicking a Varroa. Yeah, so there we are. So these are the different scenarios we identified that we wanted to test. So we obviously had to have a pot where we set up the mites on the brood. That's our control. We know they survive on brood, so that's our positive control. And then we started thinking about all the other likely scenarios from a potential illegal import. So we thought, well, mites on dead bees, mites on clothing. If somebody went beekeeping in Thailand and then took their bee suit home and then went beekeeping when they got home, could they survive on clothing? We wanted to look at mites on newly emerged bees, mites on the attendants in a queen cage plus fondant, so mocking up an illegal queen cage import. Mites on comb. Maggie showed the picture right at the beginning of the Florea with the brood nest right under the honey, and could mites survive like that? And then again, mites alone, so that would then be the negative control. So how long could the mites survive in these conditions? That's it. So we set up a lot of pots. So these pots literally are deli pots. They're the kind that you would get coleslaw from at the deli counter in the supermarket. We use them all the time for experiments with cage bees in the lab. It's with a tried and tested method. You can make little holes in for air holes if you and then put mesh on them. We can make little holes in the side where you put a feeder then with some sugar syrup if you need it. So it was something that we were well practiced at doing in the lab. The thing that we weren't well practiced on was finding hundreds and hundreds of trop, and that took a long time. So we came up with we had all the different sort of treatments that we'd decided on that were shown on the previous page, and we wanted 3 replicates for each treatment. Ideally, we would have wanted 30 trop per cage. It's sort of more statistically viable. We ended up aiming for 20, and that was just for practicalities. We couldn't get enough trop on this round for what we needed. And the next, I don't think that's so that was it was an over a 12-hour day setting this up. Dan, Paul and I started, and Maggie and George were out at the apiaries. They were then haggling for frames to bring them back for us. We set it all up. It was a massive team effort. We worked really well together, but we set it all up and then got them in the incubator, which is the next slide. Yeah, there we are. And we set them so they were put in there at 33 degrees, 60% relative humidity. Again, another example of how wonderful our hosts were. They just gave us this cleaned out, virtually brand new incubator to use, and allowed us access to the lab whenever we wanted it, including weekends, because the whole point of a survival study is you've got to check them every day. So that was Saturdays and Sundays as well. So at 24-hour intervals, we then looked at each of the pots and counted dead mites and live mites. The dead mites we removed, and we've got a video here of George very carefully checking for the dead mites. We found that a jeweler's hand lens with a light in was useful as well. You can see you can see the light on the pot there. I think he's got one of the camera lights on him there just to make sure, but literally it was poke the mites and if they don't move, then we collected it out. It was obviously a lot easier to do that in that pot because that's got dead bees in. The pots with live bees were a lot more difficult because we had to go in through the feeding hole at the side, otherwise the bees would have escaped. Can I have the next slide, please? So pot study 1, lessons learned. Wow, there was a lot of lessons we learned. Obviously, if this was done in the lab in York, we would never in a million years set up a 60-pot experiment or a 48-pot experiment first time without doing a lot of preliminary work. We didn't have that luxury. We were there for 2 weeks. We wanted to do it, so we had to go kind of all in. So the first time we did it, we learnt a lot. So we learnt that the bees moved dead mites. So it was difficult to keep track of counting the dead ones. In the pots with live bees, we were kind of putting a little circle around the dead mite on the plastic from the outside, but then however carefully we put them in, we then realized the bees were moving them. The cages with fondant, the mites crawled into the fondant and got stuck. Mites crawled up the side of the pot and got stuck in the lid. They drowned. Oh yeah, mites drown in the feeder. They drowned, and mites get stuck in honey. So yes, they were difficult to handle. Again, because they were hard to catch, we didn't quite have the numbers we hoped for. So what we decided to do was repeat it, but reduce the number of treatments that we were using, because even though we had a lot of problems, it was easy to tell there was one or two of the treatments that we'd got in there that the mites just couldn't survive on. The clothing was one of them, which was reassuring. So we made some adjustments to the method. We decided to invert the pots the second time. We can coat them with something called a fluon, which is something we use in the lab for stopping ants cross the line. So we made quite a few amendments and changes, and the pot study 2 was far, far better. So like I said, based on the preliminary results of pot study 1, we selected the matrices that were most of interest. So we still had to have our positive and negative control, which were mites on brood and mites alone. And then we wanted to look at mites on dead bees. So in order to kill the bees, we put them at -20 for a short time to kill them. We didn't want them to be damaged in any way, because if there was a damage site on them, we thought, well, then that'll be a lot easier for the mites to find a feeding site. We wanted to look at the mites on the newly emerged bees. We had another go at the adult bees and fondant, but we just set it up differently to make it more difficult for the mites to get stuck in there. And we looked at mites on comb as if we'd had an illegal import of comb being intercepted. Again, so we had 3 pots per treatment, 20 mites per pot. And because pot study 1 showed us that there's an initial very fast drop off, we set it up and then counted the live mites at 1, 3, 5 and 21 hours, and then 24 hours, and every 24 hours until the mites were all dead, including the weekends. A true survival study, you do the counting at each 24-hour interval. But as I say, because there was such a fast drop off, we wanted to gather as much data as we could in case we needed it, because that was our only opportunity to gather that data. Yeah, can I have the next? So the results that we got. So the paper's in preparation, as Maggie said. So I've just got a summary of the results here because, so the mites, they die when they're on their own, which is reassuring, and they do not survive on dead bees or on comb. So that was all nicely reassuring in case there was possibly the illegal import of honeycomb. What we did find that the mites survive on larvae, as you would expect, and they survived on emerging bees. Now these, when we say emerging bees, they were literally they were just emerging, and we carefully picked them off and put them into a pot. So they were as fresh as they possibly could be. And what we found was the time point at which we got 50% death rate was similar, certainly within statistical allowances. It was similar for both the larva and the emerging bees. However, what we did find was the death rate of 50% was faster for the trop on the emerging bees rather than the larva. So it's almost like some of them die really fast, but some of them can survive quite a long time. So why is that? So one of the options we wondered is, do the mites find a feeding site and then stay there? So if one mite finds a feeding site that it can establish on the emerging bee, does it think, right, I'm staying here, I'm not moving, you know, I'm going to stay here? And that's one of the ones that can then survive a long time. Do the males or the females or the gravid females fare better on emerging bees or not? Is there a sex or a pregnancy state differentiation as to which mites can survive longer? So the good news is we've got these mites. So we had permission from DEFRA to bring them all home. They are all in the individual pots, sample tubes in the freezer. And so something I intend to do is I'll be able to look at these individually and work out whether they are male, female, or a pregnant female. I'll be honest, I just haven't had a chance yet. It's been quite a busy start to the season, foul brood wise, but that's something that they're in the freezer, that can be done. So that's really quite exciting, and it was something that we didn't expect to find, and that, as far as we're aware, isn't out there and isn't published. But yeah, the mites do survive on emerging bees. So this is us then moving to the whenever we were moving, we didn't stop working. We were that was actually us leaving Chiang Mai on the left, and how we didn't put that taxi on its axles, I don't know. We had so much stuff and equipment with us, but we did. And that was us working on the train down. It was a 10-hour train journey from Chiang Mai down to Bangkok, because we thought it'd be greener not to fly, and also we were mindful of the budget. So I think it was £6 for a 10-hour journey it worked out at. And then I think Maggie is on next, back to sexing trop.
Yeah, so Thailand wasn't all about the experiments and the research. We also wanted to improve our skills. So Kitty Fong showed us how to sex Tropilaelaps mites. And to tell the sex of a mite, you need to look at its anal shield, which is here. So that's the anal shield. So the photo on the left is a male, and he has if I can find my pointer. He has a pointy anal shield, so you can just sort of see that there, whereas this one in the middle here is a female, and they have a sort of flattened anal shield. And the other thing that Kitty Fong told us about, so this is a pregnant female, and you can see that she's quite a bit fatter than the other mite there that's not pregnant, and really they're a bit like they're sort of pop-out suitcases. So when they get pregnant, they seem to pop out, and that's part of the work that Victoria has used with all of those mites that we've collected and taken back to the UK, and that will sort of really add to the research that we're doing. So photography was one of the other objectives of the trip. So we really wanted to get good photographs and videos of Tropilaelaps, because currently there aren't really that many good high quality photos or videos. And for presentations like this and educational material, it's really important to have good quality images to show everybody what we're talking about. So I'm not sure if Dan's managed to join us yet or not, but so Dan, as well as being a beekeeper, he's a bee farmer, he's a bee inspector, but he's also a wildlife photographer, and quite frankly he is a bit of a photography nerd, especially when it comes to macro photography. There was many, so him and Paul really got into some really in-depth discussions about what lenses and lights and kits, and unfortunately I can't. Dan was going to tell you about this. I don't really know anything about it, but you can see he had some very, very fancy setups here, which were very impressive, but he did get some absolutely amazing footage, photographs, videos, and the quality of the images are really good. So, and
that's an image there that he was cursing me because I asked him to take, and that was trop floating on water, and so it moves. There's different depths. I think
the
that
light's
was
funny, and yeah.
But he got the shot. But yeah, that was something that we asked him to do.
So this is one of the videos that Dan took of some mites, and we'd marked the mites because we wanted to track the mites and see if they were setting up feeding sites. So that's something sort of that came out of the pot study, and we wanted to see if they set up a feeding site, would they stay and guard it? Now Dan took this amazing footage, but I'm the one that marked the mites, so I'm going to take credit for that. It's Tropilaelaps mites are really tiny, and I'm quite impressed with myself. It's going on my CV that I can paint bee marker paint on Tropilaelaps mites. So you can see we've got yellow, blue, and red ones there on a freshly emerged bee. They tended to hang out around the thorax there. They'd go into the gap sort of between the thorax and the abdomen where the bee sort of couldn't really groom it off themselves. And again, that's something that we'd like to look at is, are they setting up these feeding sites? Exactly what are they doing? But you can see all of the details on the bee there. You can see the hairs on its eyes. You can the level of detail there really is quite amazing. And this sort of footage just doesn't hasn't existed before. I think you can see there's a mite sort of tucked in just here and here that we suspect is feeding. So that would be something that we'd like to follow up in the future. So So the objectives, so while we're in Chiang Mai, we managed to achieve a lot of these original objectives. So the field workshop went really well, a bit of an initial fright that we couldn't find Tropilaelaps, but we pulled it together and we got on with it, and that worked out really well. We demonstrated that the old SOP was insufficient at detecting Tropilaelaps. We developed new methods to detect trop, so the uncapping using the tweezers and the icing sugar roll. We got these fantastic photos and videos. The pot experiment, so we'd had a few problems with that at the start, but we managed to resolve them, and we got some really good data about what Tropilaelaps could and couldn't survive on. We also made some great contacts while we were there. And that's really important for future work. So if Tropilaelaps were to ever arrive in the UK, we now have a whole sort of host of contacts that we can phone up if we need sort of any help. So not only was there Bom and Kittifong, we met Dr. Dominic Kamenzid and Dr. Lars Straub. So they're on the right-hand side there in this photo. They're from Bern University, and they were doing research into Apis species of bees and the viruses that they have. So Chiang Mai was pretty successful, but that wasn't the end of the trip. So Chiang Mai is in the north of Thailand here. We'd spent 2 weeks there. We travelled down to Chonburi and Burapha University, which is here. So it's about an hour and a half to the southeast of Bangkok, and also happens to be by the beach. So at Burapha University, we wanted to address the objectives that we'd originally set out to achieve, but we hadn't managed to do in Chiang Mai. And that was really looking at Tropilaelaps on its natural hosts. And we wanted to get a bit of a better understanding of how it might spread to the UK on these different species of honeybees. So this is Guntama again, our high-heeled wearing beekeeper, and her team from Burapha University. We're also joined by Jakarut Matip, so he's here, and some of his master students, and they're from King Montut University as well. So they came out and did some work with us. We spent the day in the lab with Gun, learning about the different species of honeybees in Thailand and the mites that infest them. We also gave a presentation on what we do, what FERA does, but it was during this time that we spent with Gun that we heard about Uvaroa. So prior to Thailand, I'd heard the name Uvaroa, but I didn't really know much about it. So therefore, it was quite shocking to hear Guntama talk about this mite. So it naturally infests Apis florea. So that was the mite with the honeycomb that I showed you at the start. And all 4 native, so it can also jump species to all 4 native species of honeybee in Thailand. And it's also known to transfer to Apis mellifera colonies. So Uvaroa, it reproduces more quickly than Varroa. And it also causes damage similar to Varroa, but if it were to arrive in the UK, this would be a new novel parasite, and our honeybees would have no defense against it. And it would also introduce sort of new novel viruses, and they would probably cause quite significant mortality amongst our bees. So the other thing that Gun said was that she had also come across this mite in Apis mellifera colonies in India. So obviously this is quite a bit closer to the UK than Thailand. So this is an Apis dorsata colony. These are the natural hosts of Tropilaelaps. They're a migratory bee species, so the giant Asian honeybee, and they will abandon their combs and their brood, and they move on. And they do this to follow nectar sources as different plants come into flower in different regions. But they also do this as a means of dealing with pests and mites such as Tropilaelaps, because when they migrate, they leave that comb behind. So they leave the vast majority of the parasitic mites there in that comb, and it just lowers the mite levels. So we spent a day with a honey hunter in Ratchaburi province, so that's the southwest of Bangkok, and we saw Apis dorsata. We also saw Apis cerana, Apis florea, so that's Longman, the honey hunter there. So on the left is a colony of Apis cerana that we saw in a well. And so these are the Asian honeybees. They're also cavity nesters, and beekeepers in Thailand do keep them in hives. But the colonies are smaller than Apis mellifera, and they swarm quite a bit more frequently. And again, this defense has evolved to deal with parasites. So they're naturally infested by Varroa. They also groom themselves more frequently than the Western honeybees, and they will uncap brood to stop the reproduction of Varroa. On the right, we've got Apis florea again, so the red dwarf honeybee, and this is the one that's naturally infested by U. Varroa. But all of these mites, they'll all quite happily jump onto the other species and infest them. So, and also all of these mites will also infest Apis mellifera. So the problem with our Western honeybees is that they haven't evolved any of these defense strategies that the Florea, the Serrana, the Dorsata have evolved. So Burrup University, they do a lot of research into infestation of these different mites in all of these different species of bee. And while we were out there, we just learnt so much. And Burrup was probably one of the most memorable days. It was truly amazing to sort of see Apis dorsata and Apis florea out in the wild like that. So really that was it. That was our trip. So we've only been back for a few months. We've already done quite a bit of work. There will be articles published shortly on the work we did. So we've got some going in the BBK News and Bee Farmers, so not only on the research work, but also information on Thai beekeeping, on the honey hunting day, so a bit more sort of general interest. Also keep an eye out for the scientific papers we've written on the work. These will go into quite a bit more detail on the actual research and the results of what we found out. So really though, the trip, it answered a lot of questions, and we did meet a lot of the objectives that we set out, but you know, it also generated a lot of questions that we didn't get to answer. So one of the regrets was that we didn't get to trial the UK Varroa treatments. So we also suspect that Tropilaelaps are setting up feeding sites on the freshly emerged bees, but we don't actually sort of know that sort of for sure. Kitty Fong was telling us about how Tropilaelaps mercedesi can survive in countries like Korea, where potentially they have broodless periods and these mites are overwintering. And really, we don't really know very much about that. So again, lots of questions that we haven't quite addressed and that we would like to look at in the future. And finally, just thank you again to BDI. This trip couldn't have happened without your support and without your funding. As I said, we found it incredibly valuable, and I think it's been incredibly valuable to UK beekeeping. You know, the National Bee Unit, FERA, we're certainly in a much better place now to understand what a threat Tropilaelaps sort of poses to us. Thank you to the NBU, FERA and DEFRA policy. Obviously, they let us go off to Thailand for 3 weeks. You know, they paid our wages and things like that. So that was a big help. Obviously Ying, so she isn't here, but she was amazing. She sorted out taxis for us. She sorted out equipment for us. Poor Ying, because we were working late into the evenings. So did Ying, because we were working at the weekends. So did Ying. When we had to travel down to Burrup, she was obviously left in Chiang Mai. She finished off the pot experiment for us. She was just an absolute saint. Thank you to Ariel and Max, so from Vita Pharma and Vita. So they supplied the Varroa Easy Checks and the Vita Sticky Floors, so they were really useful. Also, thank you to baby Theo. So George's partner Anna was heavily pregnant while we were in Thailand. The baby was due, I think, about a week after we got back. Quite a lot of concern that she might actually go into labour while we were there. So Theo very obligingly hung on until his dad got home. And finally, thank you all for listening. So that's the end there. And any questions?
Thank you very much, both Maggie and Victoria. Yeah, it is. I'll ask one question. We'll just have a few questions because it's moved on. Time's moved on a bit. Did you come back more concerned that Tropilaelaps is a threat to this country's bees, or less concerned that okay, you've got a plan now and we're likely to be able to deal with it?
I think probably more concerned when we came back. I think. Certainly that first day in the apiary where we just couldn't see Tropilaelaps, for me, that was really very frightening. You know, not wanting to sort of sound big-headed or anything, but as bee inspectors, all we do is we look at hives and we look for disease, and we do that minute examination of colonies. And I think we were probably a bit cocky when we went there, and we went into the Chiang Mai University apiary and we said, well, we'll go and find some Tropilaelaps, and it was just so difficult to see. And I think a lot of what we rely on in the UK, so yes, we do these inspections, yes, we have voluntary central apiaries and enhanced central apiaries, but we also rely on beekeepers to be able to detect these things and to know what they're looking for and to detect them. So personally speaking, I think I was quite sort of frightened that they were just so hard to see, and that if they do get it, just how are we going to work with them, and how are we going to monitor them and detect them and deal with them. So but Victoria, I don't know how you feel.
I agree with everything Maggie said, but from my own personal point of view, I feel happier now, because I know now that as a result of adaptations we've made in lab protocols after going to Thailand, if it's there, we will find it in the sample. Whereas before, the methods, you know, they've certainly, I think a polite way of saying is suboptimal. But we've made amendments now to both how things have been done in the field, so consequently differences to the samples I will receive in the lab. So I'm happy. Oh, I've just seen Dan's here.
Good to see Dan.
Yeah. I was just going to say, Dan, we've watched all your videos, Dan. Thank you very much, and seen your close-up photographs.
Yeah. So I think it's fair to say, exactly as Maggie said, you know, there's more questions, a lot more questions about, you know, spotting in the field, but I think now in the lab, you know, we've got adaptations that are hugely beneficial.
I think one of the questions is as well is you know, is really looking at where do we go, you know, if Victoria does get a positive, what happens next? You know, that's probably one of the you know, we've got a contingency plan, and you know, maybe that's something we need to dissect slightly more, but that's, from my point of view, quite an interesting area that maybe, you know, we can think about exploring too.
I think
Maggie said we didn't get a chance to do the treatments for surveillance and all the rest of it. So there's
anybody else got any questions they'd like to pop up and ask?
So could I just say one more thing? The previous question, I think, as you mentioned earlier, Martin, you know, the lack of bee suits and everything there, the bees were so placid, so easygoing, you know, to really experienced beekeepers such as Maggie and Dan, you know, George and Paul, they'd never seen anything like it. You know, perhaps the temptation might be there for someone to bring something back that they shouldn't, an illegal queen. You know, it almost the gentle nature of the bees could turn out to be problematic.
Okay. Anybody got a question they'd like to ask? No, you've answered all the questions that they had, ladies, and Dan brief. Well, Dan's photographs and your video. I think your videos are very impressive. Okay. Thank you very much, all of you, for your talk and your good quality stuff. We learn as we go. So yeah, thank you, and I'm sure we'll see you all again. So
thank you.
With that, I will close the meeting. Thanks, guys. Good night.
Good night. Thanks for an interesting talk.
You're welcome.
Thank you. Bye.